Yeah, hi, welcome to this workshop. My name is Hannah. I am a PhD student in the lab of Knut Drescher, and I will be giving this workshop today, which I made for the I2K, the Images to Knowledge conference in 2022. However, this will be available long after, hopefully, so that I hope that even people who weren't able to join the I2K will be able to benefit from this workshop, which focuses on BiofilmQ. BiofilmQ is the software that enables us to perform community analysis of microbial communities, so quantification of microbial communities. And in particular, you may have guessed from the name biofilms. At the I2K, Niklas, Eva, and I will be available for questions, and we're also available for questions after that through the forum, the image.sc forum that I'll mention along the introduction that I'm doing right now. So I want to start with a brief intro into biofilms in general to give you an idea of why we wanted and needed to develop the software. Biofilms can be described as a three-dimensional structure of cells embedded in an extracellular matrix, which can, for example, look very simple, like this pile of cells, basically, that you see in this image here. Biofilms are currently considered to be the most abundant form of microbial life, and as such, they are incredibly heterogeneous. You can find biofilms in all kinds of sizes. These are just two examples, going from a very small biofilm that is really just a couple thousand cells to an incredibly large biofilm on the microscopic scale. So you could see this with your own eyes and see the beautiful wrinkling pattern that is emerging here. But there are lots and lots of other types of biofilms, environments in which biofilms grow, and there is really a lot of diversity between them. These are all example images of biofilms that we have at some point analyzed in the lab, and you can see that they are very different in terms of the shapes of the biofilms, in terms of like also the questions that you would ask when looking at this. And of course, they have different sort of scales that you can see here. And there's not only diversity between them, but even within a biofilm, you can see this macrocolony or with this other macrocolony, that there is, even within a biofilm, you have a sort of spatial gradients, you have a spatial component that you cannot neglect. So we were performing analysis, quantification on these different types of images, and we found that we kept on sort of reusing the same code, but also modifying it. And we were wondering if there was sort of a universal way to analyze these communities, and not only make it accessible to us, but actually also to our collaborators who asked us to do the analysis in the first place. Was there anything we could do to kind of bring the analysis to the biologists who know the questions better than us, who know the systems better than us? And the answer was BiofilmQ, which I developed together with two of my colleagues, Raimo and Eric, that you can see up here. So we eventually published this paper in 2021. However, BiofilmQ has been available since, I think, summer of 2019, and has been used ever since. It's a software that has a graphical user interface, so there's actually no programming required to use it. This was very important to us because we wanted to have it accessible to as many people as possible, and we didn't want to have that barrier of people having to learn how to program before they will be able to use it. BiofilmQ is freely available. You can download it from our webpage, which I'm going to show you in a second. And in fact, let me show you right now. So yeah, if you Google BiofilmQ, this is what comes up. We have at the top here this documentation that we made, which I strongly encourage you to check out at some point. It is really extensive. It contains so much information, and basically anything you need to get started with BiofilmQ. Then, of course, we have the website that also contains a lot of information and like all of the important links to the different resources. And then, of course, if you want to read the paper, that would be the third link in this list here, where you can just read up on what we did back when we published it. If you go to the website, it will somewhat look like this here, and you can see there are a bunch of links here at the top, which include, for example, this download link that brings you to the documentation page, where you can download different versions of BiofilmQ. One would be the zip version. This will be kind of a folder with MATLAB scripts that you can run when you have MATLAB. There's also an EXE version that you can run if you don't have access to MATLAB, because we know that MATLAB is not accessible to everyone. However, if you do have access to MATLAB, we always recommend you use the zip version because that makes it so much easier for you to make changes or to kind of make small additions if you want to, as opposed to the EXIF file. Then we have this tutorial series, which is constantly extended and will be also extended by the video that I'm doing right now. That contains a lot of interesting tutorials on how to do different things with BiofilmQ, how to get it to run. And if you encounter any questions, the forum is the place to go. So this is the image.se forum that I mentioned. Me and my colleagues are kind of constantly looking if there's a new thread there, people asking questions, and really, really try hard to always find a solution for their problems. And of course, you can also just browse through the topics that are already there, which might contain some interesting information for you as well. So yeah, let me talk a little bit more about BiofilmQ. And I'll start by talking about this kind of general workflow that we have that basically consists of 3 steps. The first one would be the segmentation, where we distinguish cells from background. We perform this cubing, which I'm going to talk about on the next slide. After segmentation, we perform analysis, where we identify the properties that we want to measure. We kind of check them in this list here, and then we can run the parameter calculation. And then finally, we also have a visualization tool directly integrated into BiofilmQ to enable you to look at your data and to see directly what happens. For example, if you make some changes to the settings here, how does it affect the outcome of the analysis? How sensitive is it? How can you optimize it, and so on? So this is all of it is sort of integrated into BiofilmQ. Starting with the segmentation, to go into a little bit more detail, what actually happens in there? Well, the first part is a very simple foreground-background segmentation where we filter the image and then perform thresholding. And this basically just tells us where do we have cells, where do we have background. What we do afterwards is slicing the structure into cubes. And the reason why we do this is that we want to have a spatial context. I talked before about the spatial heterogeneity that can exist within biofilms. So we need to have a spatial context, but at the same time, sort of the natural thing that you would be single cell segmentation. But this is not always available to us, especially if we have very large biofilms, macroscopic biofilms. You don't actually have single cells that you could segment, so you can't do this. And this cubing is really like a neat solution to this problem. And it is just really versatile. You can literally apply it to any kind of community that you encounter. If you do, however, have a very good single cell resolution, or you have a good segmentation from a third-party tool, you are free to import the segmentation and then use sort of the subsequent steps of BiofilmQ. You do not actually have to use this part of the segmentation of BiofilmQ. You can directly import your preferred segmentation. After you have segmented the cells, we want to kind of calculate properties. And these properties can be quite diverse. One thing that people often want to measure are fluorescent signals and where and when they switch on during the experiment. And this when and where is sort of summarized in these heat maps that you see here on the right. So this is called a chymograph. It has the dimensions time here at the x-axis and space at the y-axis. And in this case, space is sort of a distance to the biofilm center going from inside, or biofilm surface inside to outside. So this would be the inside of the biofilm. This would be the outside of the biofilm. And then the color indicates whatever we measure here. So for example, in the case of the matrix reporter, you can see that it switches on relatively late during development and only in the outer regions of the biofilm. I can look at architectural properties too. For example, the local volume fraction telling me where in the biofilm my cells are particularly dense. And this, for example, in this particular case, we found that the volume fraction is relatively high in the core of the biofilm at late time points. There are other properties that you can measure, especially if you have several channels. You might be interested in the interactions between those channels in the sense of how they are related to each other. Here, for example, we were doing an experiment where we had a biofilm that was invaded by another species, which is represented here in blue. And the cells were previously treated with antibiotics, which is why they're so sparse here. And we could see basically that the cells predominantly are sort of existing in the outer region of this previous biofilm. So you can sort of see how, in what areas do these invading cells accumulate, as an example. With the same type of analysis, you could, of course, also do species composition, and we can also calculate global properties, such as, for example, the biovolume. You might know roughness coefficients from the program COMSTAT, number of microcolonies, and so on. So all of these things you can measure basically from one image, which is not localized, but a global property. You can also measure with BiofilmQ. And then finally, we also have the possibility to do a high throughput analysis, a batch processing that enables you to run the same type of analysis on a bunch of folders and basically just let it running over Christmas and come back and have it done. Yeah, the visualization part is the last step of this workflow. And here I just want to mention that we have a variety of different plot types that you can perform, where you are basically free to choose the axis. And this gives you a lot of freedom to look at your data. One thing that we have already discussed is this heat map or kymograph over here, which we like to use to visualize spatiotemporal processes. Then we can have histogram or 1.5D histogram, which is one parameter resolved by another to see if there's any kind of dependency. And then we can also have scatter plots in 2D, 3D, or we call it 4D because it has the color in addition to the 3 dimensions. So this just gives you a lot of possibility to explore your data. Yeah, what are we going to do today? We are going to have a practical part following this presentation of mine. And in this practical part, we are going to work with 2 different data sets. The first data set is sort of our standard data set that is also available on the web page, where we have these sort of 2 biofilms, so to say, that kind of grow next to each other. We have 2 channels, a red and a green channel, and we can see that all of this yellow part here is actually cells that are both red and green. So we can see that we have 2 subpopulations here. One is like the lower biofilm, which is only red, and the other biofilm, which is both red and green. And we are going to look at this data set as an example, and I'm going to show you how to analyze it in terms of image alignment, in terms of segmentation, what type of properties you might want to measure. And then interestingly, or in this particular case, what might be interesting is also to figure out how can we separate these 2 subpopulations to compare them to each other, for example, in terms of growth rate or any other property that you might be interested in. And this sort of workflow is kind of a quick compilation of many of the tutorials that we already have on YouTube. So it's a bit of a quick start. If you want to learn more about some of the processes that we do in detail, that I'm going through a little bit too fast, in your opinion, you can always check out the tutorials and go back to them. So this is just to give you a very brief, but hopefully somewhat comprehensive overview of what you can do with BiofilmQ with this one example. And then we have the second data set, which looks like this, where we have a macroscopic colony, also with 2 channels that represent 2 strains in this case. And it's sort of a competition experiment where we have spotted both of them in the center, and then they kind of grow outwards. And we are interested in the question, which one of them is kind of becoming more or less abundant as time progresses, and time progresses across basically this spatial dimension here. So this example will be particularly interesting for those of you who are interested in the merging capabilities of BiofilmQ, and we found that there are actually quite a few users who are kind of interested in this merging capability and who might be interested in using that. And then, of course, this will also show you what options you have to look at things in a spatial context, such as this sort of time progressing along this side. So yeah, we will have these 2 data sets to look at in the practical part of the course, which is going to start right now. And I hope that you enjoy it. Yeah, so we will dive right into using BiofilmQ in this practical example, the very first example that we're going to do today. And what I have brought up here is MATLAB, where I have opened the folder that you can download from our webpage. So this is now opening this BiofilmQ folder, in which you can see, importantly, the biofilmq.m file and the vig file. So this is what it should look like for you at home as well. We can start BiofilmQ now by running this MATLAB file, biofilmQ file. And while it is opening in the background, we can already have a look at the data that we're going to work with in a moment. So this is the data that you can also download from our webpage, download from the documentation, and it contains images in 2 channels and for 7 time points. You can see that the naming convention of these files already tells you something about these channels and time points. So the time point is given as a frame number here in the back, ranging from 1 to 7, while the channel is given with the CH and then the channel number indicator. It's important that the files are named this way for BiofilmQ in order to recognize which frame it is and which channel it is. But as you import your data into BiofilmQ, these indicators will be given automatically. The same way, you will also automatically generate these metadata files, so you won't have to worry about them. This is just some data that BiofilmQ needs in the background to know how to handle your images. As BiofilmQ has started, we will hopefully in a moment be able to see the start screen and to choose our directory. So I hope that you can see and read everything that I'm doing here in terms of resolution. If you want to open an experiment folder, you have this button up here called Browse, which you can use to move to your directory. So in this case, for example, I'm actually not in the right folder. I have to go to the 2022 version, into data, and here is this sample data folder that I have shown you just before. So we just select the folder and then we press on select folder. BiofilmQ will now read this folder and it will read the images in there and the metadata, and it will display the information that it has seen. So now we can have a look at this overall BiofilmQ view. And if you've run BiofilmQ on your computer before, you will kind of be hopefully a bit familiar with this, but I will anyway walk you through some of the areas that you see in here that might be of importance, or that are of importance to us, starting with this file list over here. So here you can see these 7 files, and you can see also that it has selected a channel automatically. So we have selected channel 1. We are looking at channel 1 here. Also, you can see that the first image is selected, and this image is now given as a sort of representation, and this is a set sum representation here in the center of the view of BiofilmQ. When I shift to a different image, this image in the center will also change. I can switch between channels by switching this fluorescence channel drop-down menu down here. So I can go to the second channel and have a look at it. And you can see in this case, for example, here we have a much darker signal apart from a few cells that are still bright. And you can also see, and this is something that was mentioned in the introduction, that only one of these 2 biofilms that we saw before actually carry the signal of the second channel. You can display the channels in a so-called merged view, which is a good way to get an overview of how your signals interact with each other, how the different channels interact with each other. So here we can see both of the biofilms. We can see the red color represents the first channel. The green color represents the second channel, so we can see all of this kind of yellow area. This would be cells that are both fluorescent in the green and in the red channel. And then there is this area here where we have cells that are only present in the red channel. I will move now back to channel number 1, because that is the channel that we want to focus on, that we want to perform segmentation on. Because in BiofilmQ, it is important that you are always capable of doing some kind of segmentation. So it's good to have a continuous signal, such as the one here, where every cell just carries this one signal that can be used for segmentation. And then the second channel can be used, for example, to distinguish cells that belong to these 2 different subpopulations. So this is how we can look at our data in this image preview here in the center. We will look at it in some different ways, and we'll come back to that over and over again through the workshop, because this is actually the area to look at if you want to have any kind of visualization of your data. You can see here on the side that we actually have the option to also look at our images, not just as a Z projection, but also, for example, in this auto view here, or as a Z stack. So if I look at this image as a Z stack, a window will open in which I can basically scroll through the data and see how the cells are fluorescent in the different layers. So moving now to this area over here, which is where you can perform your calculations, your segmentations, and so on. And you can see that we basically have this step-wise approach where you start with some preparation, you then perform segmentation, parameter calculation, and so on. And it is recommended that you do these steps in this order, but you can, of course, also skip some of them if you don't need them. For example, in preparation might contain some things that you do not want to do. And if you want to have a very detailed view of kind of all of these 5 different steps, you can also look at our other tutorials online. Today we will focus on the things that we want to do for this data set, the things that are relevant for this data set. And this is, for example, image alignment. What is image alignment? Well, sometimes you can observe, and I'm not sure how well, yeah, I think you can see it very much between the first and the second frame here, that we have a shift between these 2 time frames. So apparently something has happened, maybe to the stage of the microscope, to the biofilm. It has shifted a little bit. And this can mess with some of the analysis. Usually it's not a huge problem, but sometimes it can be important that we really have everything aligned perfectly. And in that case, you want to perform image alignment or registration. In order to do this, we need to set a reference frame. And the reference frame will be basically the frame to which all of the other images are going to be aligned to, which is why it makes sense to choose the last frame of the time series, because this is where the biofilm is the largest, and this is what we want to have sort of in the center of our field of view. So I can set the current frame now. I can decide whether or not I also want to align everything in Z in case that there was like a small shift in that direction too. And then I basically just click on register images. Before I do that, I should check that the image range that you can see up here is the way that you want it to. So in this case, you want to perform the registration for all of the 7 images. But it's possible, for example, that you want to perform a certain type of analysis step only for 1 image, in which you can, for example, select that image and click on this button. You can also select several images at once, where you can see here now how you can change this sort of order and how you can run a part of the analysis only on a subset of the images. But we want to do this for all images, so we will click all images and then we will click on register images. And during this time that BiofilmQ registers the images or aligns the images, it will also actually tell us a little bit how the movement is between one frame and the next. And here in the background, we can see that BiofilmQ sort of overlays 2 time points, both in X, Y, and in the Z direction, or the XZ, I think it is then. To kind of give us an overview of what is happening while the registration is performed. So we can see how the biofilm has moved. Just a couple of pixels, actually really not that much from one frame to the next. And here we get this information now, registration is finished and the window may be closed. So this is actually quite fast. So now we already have our data aligned. And you can see at the bottom here that we can choose to display the aligned version of the data or the unaligned, the original version. And this basically is shifted by this very small area. Yeah, so now that we have done this registration part, we will move on to the segmentation. And the segmentation is probably the heart of BiofilmQ. There are a couple of tutorials online on YouTube where you can look at the segmentation and the different steps that you do, the impact that, for example, different filters have. And we will also do this for our example data here. One of the first things that you have to choose if you want to have a cropping or not. And in our case, we probably don't need a cropping, even though it might be of use to have only a part of the image. This basically just saves us time in this case. So I can say, well, biofilms are only in this bottom part here, so maybe I just want to perform the segmentation for this bottom part. But of course, you could also think about selecting one of the 2 biofilms by a cropping and then performing your analysis only on this biofilm, and this will save you time, and it will also get rid of some of the background cells that you are not interested in, maybe the ones up here, for example. We can perform pre-processing, which in this case is also not necessary. This would be, for example, to invert the stack. because BiofilmQ assumes that your biofilms are basically growing from bottom to top. However, some people have a different type of microscope where the image would just be inverted, and then you can correct for this. You can also correct for a tilted cover slide, and you can scale your image up or down. The scaling can be useful if you have very large images and you find that they take very long to process in BiofilmQ, in which case you might just want to scale them down by a factor of 2 or whatever. So then you would just put 0.5 here to scale them down by a factor of 2, and this is going to happen in both the X and the Y dimension. You can also upscale them to have a finer sort of segmentation process and like a finer process afterwards, but this will not actually increase the information that you have in the image. It's just basically going to replicate pixels. In the denoising part, you can choose which filters to apply before you perform the segmentation. And here it's really important that you play around a little bit with the different filters that are here and with the settings for these filters, and you choose which ones work best for your example. There is really not a one-fits-all solution, so you do have to kind of think a little bit about what you want. Look at what the result is, and I'm going to show you in a minute how you can look at the result. So here we, for example, have the option to denoise our images by convolution, which basically just means that we blur the images a little bit, which gets us rid of salt and pepper noise and some of the fine noise that might be in the image. Here you can choose the size in X, Y that the filter is going to average over and in Z. We can also perform a median filtering along Z, which is useful if you have single cell resolution and you have some floating cells that are shifting between the images, and you can sort of get rid of them. Those would be cells that are not actually part of the biofilm. And then there is this top hat filter, which serves to essentially darken the areas between two cells. So this is really useful if you have a single cell resolution. Usually, if you don't have single cell resolution, I would not recommend that you use it. But how can you actually know how these filters apply to your image, what it looks like after applying these filters to your image? Well, this is something that you can find out in this part here, where you have the thresholding, and you can actually see how the thresholding affects the image, but you can also see how the filters affect the image. So first, at the top here, you choose the thresholding method that you want to apply, and we have different ones implemented here. One of them is also very important, the manual thresholding, where you just pick a number, and you don't have to pick it just like a random number such as here, but you can actually pick it visually, which I'm going to show you in a moment. But you can also use, for example, auto thresholding, in which case you can choose between having either 2 or 3 classes. And in a 3-class case, of course, you can choose whether the second class, the middle class, is going to be foreground or background. The sensitivity is a factor that will be multiplied with the threshold that is chosen. So if you find that your images look like they're always a little bit too high thresholded or too low, you can correct for that with the sensitivity value here. And this is also what will happen if you press this button here. You will choose the sensitivity value in case that you have picked an automatic method here. And in the case of a manual thresholding, you will choose your manual threshold. So what does this button down here do? And this is actually a very important button. So just to quickly discuss, we have this drop-down menu here. We can choose whether we want to perform our sort of visual thresholding on the raw image or after image processing, where image processing means that the filters will be applied. And that means that we will be able to see also what the filters do. So we are going to choose after image processing in this case, so that we can see the effect of the filters and we actually see what the outcome would be like afterwards. So now we will click this button here, and a window will open, and this may take a moment because we're actually applying the top hat filter, which uses the, I think you can have a look at this here. Yes, so it uses the parallel pool of your computer, which is actually quite useful if you have several cores. If you have a strong computer, because then things will just run much faster, but waking up the school typically takes a while, which is why we will be waiting for a short moment here until my parallel pool has woken and the calculations can be performed. By the way, I'm doing all of this on a laptop that is approaching its third year. being used. So I really hope that this will be a realistic representation of what running BiofilmQ might look like for some of you in case that you do not have very strong computers available. Yeah, so now we have opened this window here. And let me explain to you what you see in here. So we have the cells that appear as grayscale against the background, which is shown in blue. This is so that you can better distinguish with the current threshold which part of your image would be background and which part would be cell. The threshold is displayed down here, so you can adapt it, for example, by putting in a different number. Yeah, just put 1,000, or you can use the slider to change the value. And the value will still be displayed there. And this really helps you to get a feeling for where you want to set your thresholds. I can start with a very low one and then just go up, up, up until I'm satisfied. So this is where I can see the threshold, and if I'm happy with my result, I can close this window, and you will see that the threshold we have here, 586 something, will be now written in here. But the other thing that I wanted to show you is the effect of the filters, right? So you remember what the image looked like before. What happens if we, for example, apply a much larger window of averaging? So if I do 11 and 3 here, for example, let's have a look at the effect that this has on the image. And now the same calculations will be run again, and we will get the same sort of visual feedback. But this time the filter that was applied is a filter that had this other thresholding image value. And I hope you can see it here, but actually let me bring up the other one again as well, just so that you have it for comparison, what the difference is between those two images. So I can actually click this button several times, even though it says please wait. And it will open these thresholding images so that I can actually put them side by side to pick which conditions fit best for me. Another nice feature is that these windows are actually synchronized as well. So you can see as I scroll through here, in one window, the other window also changes. And now I think you can see the effect that this filter had. So you can see here on the right, this is the original filter that we had with 5 as the sort of size of the filter. And here we have 11 as the size of the filter, and you can see that the cells are much blurrier here, and it might be harder to distinguish between them if you want to have a very fine approach at identifying the outlines of the cells. Depending on what you want to do, one way or the other can be better for your use. For example, if you apply like a large averaging filter, maybe combined with a relatively low threshold, you will be able to close gaps in the biofilm if that is your solution to your problem, or can be a solution to your problem. Whereas if you have a filter which is not so much averaged, you might be able to distinguish cells better. And the same goes for the top hat filter. And now I'm going to not apply the top hat filter, which in this case, I'm not sure if it's going to make a large difference or not, but let's have a look at it. So here we do have to adjust the threshold quite a bit. It shows the one that we had before. And let's go to somewhere in the middle of the biofilm, and I'm going to put next to it our previous version where we did apply the top hat filter. So I think actually we can sort of see it's a little bit difficult to compare the two because we have different thresholds that don't match up perfectly. But I think that what you can see is, for example, that this area here, the cells are much more clumped together if we don't apply the top hat filter. So this is how you can basically just giving you this example of how we can go through your data, how we can apply, not apply filters, look at them side by side and choose whichever one works best for you. So now we are going to choose whichever one works best for us. And I would suggest that we apply the top hat filter, and maybe we can do an automatic threshold using also with 2 classes. Let's have a look at how it works out in this particular case, if we need to adjust it somehow, if it just works relatively well out of the box. So this is what is basically suggested for us. Let's have a look at that. Personally, I would say that this threshold is too high, so I'm going to lower the sensitivity here to a slightly smaller value, maybe even smaller, to get to a point where I feel like we actually see everything in the biofilm. And in this case, of course, you can see that the cells are not like distinguished very well, but maybe that's not my main concern at this moment. You will see in a moment why this is not my main concern. So here, this is now the thresholding that I would suggest. You can see all of the cells. I mean, we can go a little bit higher, but in principle, this is where I would roughly go. And you can also see that the cells, they are clumped together. You will not have a single cell sort of segmentation with this approach. But let's keep it this way for now, and let's move to the next step, which would be the objects declumping. And this is why I said before, it's not actually that important for us, at least for our usage in this case, for these images, that we do have a single cell segmentation, because we are going to dissect them anyway. If you do not want to dissect them, if you have a good single cell segmentation just by the thresholding, for example, you have a very good image, or you have been able to find perfect parameters that give you a good single cell segmentation, then please use this none option here, where there is no dissection going to be applied. Or if you have a third-party tool that gives you a perfect or a good single cell segmentation, you can import this with a label image. In our case, we are going to use cubes to dissect our biofilm, and this means that we will have a spatial resolution without having the need for single cell resolution. We can pick the size of our cube length here, and you can see suggested is 20. That's just the default, but we could put something else. We could make them smaller, we could make them larger. And the rule of thumb is essentially you want to have them as large as possible to save processing power, but as small as necessary. And if you think of it, depending on what is the kind of structure that you want to still be able to resolve. If you look at this, for example, I might want to be able to resolve sort of single cells. So maybe it's best that I keep it this way. Maybe I even want to go lower and say, okay, but then my cube size needs to be lower than the size of a single cell. There might be other cases where you say, I'm not actually interested in these single cells. Maybe I can go even higher. I can go to 40. And then this is going to be your resolution at which you can basically see different phenotypes in your biofilm. And this value down here essentially represents the side length of such a cube. So in our case, I think I'm going to choose something like 15 or 16, something that is close to 1 micrometer, which is roughly the size of the width of a single cell. Right, so now I have my dissection method. I can do some post-processing, in which case I can, for example, and this is actually quite useful, remove small objects which have been identified with the thresholding, but they are really, really small and therefore most likely noise. Here we have the size of 100 voxel. We can also make it 500, because as you can see at the back, this is actually still a very small object. The other options here are doing a 3D median of the binary image, so that if a cell only appears in one slice, it's not actually considered to be a cell, because it might also be a floating cell, similar to this part here. And we can choose, and that is if you have a very long time series and you have a lot of cells, and you know that essentially after, I don't know, 6,000 cells, your resolution is going to be pretty bad and your segmentation is going to be pretty bad. You can also choose that you want to finish the processing after a frame that contains a certain number of objects. So this can be helpful to save time if something went wrong with the segmentation, that you just move on to the next folder. But in our case, I think we won't need to worry about that, because that doesn't look like 6,000 cells. All right, the merge and transfer is something that we are going to discuss a little bit more in the second part of the workshop. So I'm going to skip over this here. This is not something that is actually performed during segmentation, but will be performed after segmentation. So now we are going to segment the cells with these settings that we have chosen. And I expect that this is going to take a moment, even though, in fact, I think it's not actually that that's low. So here, if you are getting bored in between and you're going to get your coffee, come back, you can always see here what has been happening. In the meantime, we can see the different steps that BiofilmQ has performed. Over time, we can see which imaging is being processed, image is being processed right now. For example, right now the top hat filter is applied. After we have done the average filter, then you can see the thresholding will be done and the different steps of segmentation are performed. And this will now be applied on all of the 7 images. Yeah, so now the segmentation is complete. And we can have a look at the segmentation results to check if it has worked out well. And in order to do that, we are going back to the central view here, where we have different buttons that we haven't applied yet. One of them is the auto view, and this actually has nothing to do with segmentation, but because I skipped it before, I am going to apply it now, just to show you what you would see in here. So the auto view is a view in which you can see each plane. And I'm scrolling through the planes with my mouse wheel. And then you also have the XZ and the XY projection. And these are projection of these 2 green thin lines. And you can see that when I move my mouse and I click somewhere else, these lines change and therefore the images changes as well. So we can see different ways to look at the data. And this is a way that sort of very nicely represents this 3-dimensionality of the biofilm. So to check the segmentation, I can use this overlay here. And typically this takes a moment, which is why I actually already applied it to our image here. I'm going to show you a faster version to look at the segmentation in a moment. But if you click the overlay button, what you get is this view here, which is similar to the one we have seen before. We also have this sort of 3-dimensionality. But what we have here is that overlaid with the grayscale image, we have these red lines, which indicates the outlines of cubes. And you can see now this cubing structure in here. You can see how the cells are cut into cubes, and you can see the outline of every single identified object. And this tells us now where we have cells identified as opposed to background, and we can have a look at it and go through it. And for example, in this case, decide that we're actually quite happy with the result. And we can move on with our analysis. A slightly faster way to look at these segmentation results is by clicking this button here, Load Segmentation Results, after selecting an image. You can see that this opens a sort of table down here, and I'm just going to scroll down so you can see it fully, in which I can select different properties. And these are the properties that are already calculated for each of my cubes that are sort of, the moment I do the segmentation, these are automatically calculated. And those include, for example, the volume of the cube. It also includes the coordinates, the surface of the cube, and its volume fraction. I can look at these cubes now also with an ortho view. And this gives me something, again, very similar. But here the color now indicates this particular property that I have selected, in this case, the volume. And now you can see how the cells are, you can also see essentially the outline of the cells, because you can see the cells as the background is black, which means that you can also see the segmentation results. And in addition, you can see how a certain property is sort of linked to the location of the cube. With this in mind, we can now move to the parameter calculation. And of course, the first thing we have to ask ourselves when we perform the parameter calculation is, what do we actually want to measure? There are several things that you can measure with BiofilmQ, and there are several ways to get to the bottom of a research question, but of course, they are not equally useful sometimes. And in particular, if I'm thinking about this type of data, one thing that really comes to mind is, how could I separate these 2 types of population? So I have performed my segmentation in this channel 1, which was a channel where all cells are fluorescent, but I know that there is a second channel in which the cells are partly fluorescent and partly not fluorescent. How can I identify the cells that are partly fluorescent so that I can separate these 2 subpopulations and compare, for example, their architecture? I can compare the growth rate and so on. And this is something where, of course, I want to measure the fluorescence intensity of the second channel. And fluorescence properties is one of the kind of biggest modules of this parameter calculation. So I'll take a short moment to just talk about what we see down here. So this is where we kind of place the settings for this fluorescence property calculation. I have this drop-down menu here where I can select what type of measurement I want to make. For example, I can just look at the mean intensity per object. That makes a lot of sense. I can also integrate the intensity per object. And here, in this case, an object is a cube. I can have intensity ratios of the two channels, which can sometimes make sense. I can calculate intensities per object shell, so surrounding an object. And then I can also calculate correlation coefficients. I can calculate 3D overlaps between channels if I have segmented both channels. And this is actually something that might also be interesting here. If I do choose to segment the second channel, I can actually do this 3D overlap in order to figure out which cells are present in both channels and which cells are only present in one channel. In our case, it might be enough to actually just calculate the mean intensity per object on the second channel, channel number 2. And if I want to choose this task and sort of add it to my list of things that are going to be calculated, I'm just going to click on add here, and you can see that it appears in the list. And if I wanted to look, for example, at the intensity ratio between channel 2 and 1, I could just also add that to the list. And here you can see my settings for this. And in particular, you can also see sort of these checkboxes here, which are whether or not I want to apply my filters before calculating the property. And we will choose not to do this in this case so that we are working with the raw data. Active indicates whether or not this task will be processed. So I can actually keep it in the list, but I'll just kind of inactivate it if I don't want to calculate it, or I don't want to calculate it again in case that I added something. Binary input tells me whether or not this other channel, the second channel, has to be segmented or not. In our case, it doesn't have to be segmented. We also actually didn't segment it, so we wouldn't be able to choose a task that has binary here. And then per stack means that I will have either a global or a local value. And I will talk about this difference between global values and local values in a bit. But for now, what we need to know is that a global value is where I have one value that is valid for the entire biofilm. Something as the biofilm volume, for example, would be a global value. Whereas, for example, an object volume is a single object property. So here now I have kind of selected what I want to calculate. I will check this checkbox here. But let's see what else we can do in this sort of list. We can do some filtering, removing objects that we don't want to look at, which might not be so interesting. Surface properties might be interesting in our case, and we will just kind of for the sake of trying it out, we will calculate it. Here we have to choose a range, which is sort of the surface smoothing, which I will put at 10 pixels for now, but maybe I can also just use, I think 10 is actually fine, or maybe we'll put 8, which is half of the cube size. So that sort of makes sense. Then we can calculate global biofilm properties, and we should do that just to kind of show you what they look like. We can calculate these spatial properties, such as distance to center of the biofilm, distance to the surface, distance to the nearest neighbor in a different channel. So that also requires that I have 2 channels segmented. So I couldn't do that, but in case that I did have the 2 segmented, I could, for example, ask for each of the red cells, how close is it to the kind of closest green cell? And this distance would be 0, of course, if the same is both the cell is both red and green. I can calculate a local density, which I will actually also check here. And then we will come to this in the second part of the workshop. I can think about custom parameters where I can choose sort of a combination of 2 properties that I have measured, or I can actually write my own script about them. And I think for both of them, we should also have tutorials online in case you want to look at this in more detail. In our case, looking at the distance to the center of the biofilm does not make too much sense because I sort of have 2 biofilms. But we can look at the distance to the surface of the biofilm. But again, I need to have a smoothing range that I'm going to set to 8 pixels. And now I need to also check it. All right, so now I have chosen my list of properties to calculate, and I can just press this calculate button, which is here at the top. to calculate these properties for all of my 7 objects. And depending on what type of parameters I selected and how many of them I selected, this may take a moment. After the parameter calculation has finished, we can also have a look at the parameters. And here we can do the same trick that we did before for the segmentation, where we load the segmentation results. And then we can choose to view one of the properties that we have just measured in this auto view. So for example, I can look at the distance to the surface in the auto view, and I can see that with this resolution, the smoothing resolution that I chose, most of the cells are actually considered to be close to the surface or at the surface, while only some cells deep inside the biofilm are not in contact with the surface. And this is partially because we have a lot of gaps between the cells. Another property that we can, for example, look at is the mean of intensity channel 2 that we can use to distinguish the different subpopulations. And here we can very clearly see the difference between these 2 biofilms in terms of color, where in this case, of course, the color indicates the mean intensity. And we can see that there are some areas in this other biofilm where we do have cells of the opposite color, and we can actually see that this is also the case for our original image. So this is not like a problem with the segmentation. And now if we, of course, picked this threshold between kind of the sort of light blue and the yellow greenish parts, we would be able to distinguish these 2 subpopulations. And this is what we will do in a moment when we have a look at the visualization. Let's have a look at other properties that we measured. For example, the local thickness might be interesting, where I can see that this is calculated per pillar, where a pillar is basically just one kind of straight line through the biofilm. And this basically just shows us how high this particular pillar is. And I can look at the roughness, but I can also, for example, look at this local density, which can be quite interesting. And in this case, actually, we can see that the local density is considered to be 1 almost everywhere, which means that most cells are surrounded by cells in their immediate vicinity. Now I can have a look at this visualization part of BiofilmQ. If I have calculated all of the properties that I'm interested in, of course, you could also go back and calculate more of them. But I will go to the visualization part now, and you see I just switched tabs up here. And in the visualization part, we have a lot of things to do. But what we need to focus on right now is basically just this load button, which will load the results from channel 1. And we could choose to not load all of them and limit it to up to a certain frame number. But for us, of course, we want to load the entire data set. And this may then also, depending on how large your data is or how many time points you have, take a moment. For us, it is relatively fast. Once we have loaded the data, you can see that this bottom part here has suddenly come alive. And here I can choose different types of things to look at. And in particular, I have this parameter selection area over here, where I can choose which parameters to display when I perform plotting. And I display them by adding them into this sort of axis part down here. So what I do if I, for example, want time as the x-axis, I will just click this plus here and time will appear as the x-axis. So I can, for example, look at time, and I can choose another property that I want to display over time, for example, the volume of cells. And then here I can choose a method. And this method is in particular interesting if I have some form of binning. For example, here, if I want to look at something over time, what I want as a plotting method, and this is actually the second thing I need to talk about in maybe a little bit more detail, I can, of course, choose these different types of plotting methods that were discussed in the introduction. And if I want to plot a certain property over time, what is most like suitable is this 2D XY plot per frame according to method, because the frame corresponds to the time. When I do this per frame, I basically perform a binning. I put all of the objects which are within one time frame, and I kind of put them all together, and then I can choose if I want to display, for example, the mean, and that would be then the mean volume of these objects. I can also have the mean and normalized by object volume. I can have the median, or in our case, it might be interesting to look at the sum. So basically, the entire biomass in the biofilm over time. So this is the first plot that we are going to do. And we can plot it by clicking on this plot button here, which will then open a window where I can see the growth of this biofilm over time. There are other ways to get to this particular plot, and this is something that I want to show you now. What we see here, those are the single object parameters, but I can also look at the global biofilm parameters. Global biofilm parameters means that for every time point, I only calculate one number. I, for example, only calculate one number for the biofilm volume, and this would be the one that I have actually calculated before. So I added up all of the volumes of sort of the individual objects, but I can also look at the whole biofilm volume, and this should, of course, give me a similar result. And now I'm just looking at this particular number, and you can see that this method is sort of disabled, so I cannot actually choose a method to kind of take a mean or anything, because I don't actually have a mean. I do have just these properties. I do that. I can see that essentially these two plots, or not essentially, they are the same. They are identical, because the biofilm volume is the same as the sum of the individual volumes of the objects. There are some other global properties here that I want to mention, and these are the ones that you can see down here where we have a little plus sign. And these are all the properties that exist as single cell properties or single object properties. For example, the intensity of channel 2. If I click on this plus, we have sort of an opening where we can see sort of kind of subcategories where I can say, okay, now I want to look across the whole biofilm at the mean intensity of channel 2, or at the medium or min or max or percentiles. And I can do this for all of the different properties that I have. I can just look at these means and percentiles, as well as the min and max values. And that is then across all of the objects. So this would be, for example, how I can look at this data, and how I can look, for example, at the biofilm growth over time. Now, what if I want to distinguish these subpopulations? And this is something that we have discussed before. This is why we actually calculated this mean intensity of channel 2. Now what I do is I go back to single object parameters. I choose the property by which I want to distinguish my data. And then the very first thing that I want to get is an overview of what this data looks like. So I'm going to the 1D histogram here, and I want to look at the intensity of channel 2. And as I plot this, you can see that I sort of have, and I guess if I would plot this maybe with a little better resolution, you would see that I sort of have two peaks. I have this peak here, which is sort of background and then almost melts into this low signal biofilm, and then I have the signal here. And maybe I can increase the number of bins for this histogram, which I can do here at the back, to actually see this a little bit better, that I have two populations here. Now, if I want to distinguish between them, of course, I want to set my distinguishment somewhere here, which is at about 2,100 in terms of, well, actually, it's at 509 in terms of intensity. So if I want to distinguish these two subpopulations now, and I want to distinguish them by their mean of intensity channel 2, I can use this filtering gating approach. And here is like something that I can actually not see because my screen is so small, but there's like a little plus sign. If I click this plus sign, you can see that a gating or filtering expression appears in this window. And I can now say, okay, I only want to look at cells where the intensity of channel 2 is above 510. And if I do that, let's see what happens to this previous excuse me. let's see what happens to this previous diagram that I had, where I was looking at time on the x-axis, from volume on the y-axis, and I had this 2D xy plot. So now if I do this, and actually it's plotting bars here, I should probably switch that to scatter plot. So here you can see the style of the plot. So now you can see that this looks a little bit different. And in fact, let me add this biofilm volume one more time so that the label isn't counts. I apologize for that. So there are a few bugs still in BiofilmQ as in any program. So now what I'm doing is this is basically essentially the same graph that I plotted before, but this time I'm plotting it for only objects that have intensity of greater than 510. So I'm summing up the shape volume. Let's see what this looks like. So here now I get a plot which is very similar to the one we've had before, and actually I probably should have kept the one we had before. And now what I can do, I can, as a comparison, add my next plot to this current figure. This is this button down here, this checkbox. And now I can remove my gating expression, or I can actually reverse it. And I can say, okay, but now I'm looking at the other population. So now this plot that we have here, which is blue, this is the population of cells that has an intensity level greater than 510. So that would be my bright population. And now I can perform the same plot for my sort of dark population. And I can even put the sum of them in there as well by just removing this gating expression and then plotting one more time until you can then see the sum of them. So now what we can see roughly is how this changes over time. And we can see that while the overall biomass of the sort of the low intensity population is smaller, it doesn't look like they're actually growing that much slower, or like they might not be growing slower. And then, of course, if you had more data points, you would be able to see more or less clearly, or you would be able to see clearly if or if not there is, for example, growth defect in the population that is expressing this other fluorescent protein or not. Yeah, so this is how you can distinguish between different populations. And this, of course, was just one small example of how to do this, of how to look at the data, how to visualize the data. And in the rest of the tutorial, we are going to look at the different data set where we can see some of the other features that BiofilmQ has. But for now, for this particular data set, we are finished. But this is also the data set that almost all of the tutorials that are on YouTube has been used for, which means that especially with this data set, you have a lot of other things that you can explore, that you can try out by just looking at our video tutorials. We are now starting with the second part of the workshop, in which we will work with a different data set. This different data set is actually an ND2 file, so I will demonstrate how to import this ND2 file with BiofilmQ. And we will start by choosing the appropriate directory, which is this one here, and let BiofilmQ read the file and display the information that it finds in this ND2 file. You can see that now this whole view has changed, that all of the right part no longer contains the different steps that I can do, such as segmentation, parameter calculation, but actually only has this one specific step called import to TIFF. And over here, we see that we have this one file in the folder. There were several files. I could also import several files at one. And I have some information displayed in the center, which tells me the dimensions of my image, the number of channels in the image, the number of time points, and the number of positions. This is a relatively simple file that only contains 2 channels and one image in these 2 channels. I will now import this TIFF file basically just by clicking this button over here. I could also choose to generate one experiment folder per position, but since I only have one position here, I can just import it directly. And what we can see when we go back to this directory here is that a new folder has been created. which is called macroscopic colony. And in that folder, I now have 2 images and 2 metadata files that were generated. So this is all done by BiofilmQ. As I've mentioned before, these metadata files are specific to BiofilmQ, but you don't have to touch them. They are just generated whenever you import such a file. Now BiofilmQ informs me that the export is finished, and I can either switch to the first directory in the list, which would be this one here, or I can stay in the current directory. For example, if I had several images that I wanted to import and I have only imported parts of them. But for us, it makes sense to switch directly to this newly generated directory, which contains the image. And now we can see that we are back to our basically standard type of view, where we can see this preview in the middle. We can again display the 2 different channels, and you can see that we have these 2 channels here. We have this macroscopic colony that essentially grows over time outwards. So time corresponds sort of to this dimension going outwards, and we have 2 colors. I have prepared another type of file to show you another type of import that we can do, which is not an ND2 file, but simply a TIFF file. And this is one kind of part of this stack that we are going to import here as another example. And this is actually a 2D colony, so a 2D view of this colony. And this is also to demonstrate how BiofilmQ works with 2-dimensional content. Because probably in this particular case, having the 3-dimensional content is not going to add any additional information. So actually it does make sense to only choose a particular plane, as it would be the case here. When I open this directory, I have yet another overview. And this is because here I do not have an ND2 file as before, but I actually have 2 files that are TIFF files individually. You can see that I have labeled these TIFF files in such a way that C corresponds to the channel number and Z corresponds to the set stack number, which in this case is just 1, and the channel can be channel 1 or channel 2. This helps BiofilmQ identify which image or how the images should be sort of combined into a new one. So I can now choose which parts of this file name BiofilmQ is going to use to identify channel position, time, and set position. So as we can see in the file name, the channel can be identified with an _Z. There is no time, and there is Z, which identifies the set plane. I can now try to validate my files, and it will tell me that there is no position in this file, which is true, and there's also no time in this file. So I'm just going to set both of this to 1. And you can see how here now the file has red, so both of the files are red, and they are assigned a position, a channel, a time, and a Z position. And you can see that the missing metadata that we have just filled and set to 1 are filled with red numbers. If I now click on Import Custom TIFFs, it tells me that some metadata could not be converted. This makes a lot of sense because we don't really have a time. So this is okay in that case. And again, I have this export finished dialog where I can switch to the first directory. So let's have a look how this was imported and if it was imported properly. All right, so this is my colony. This is again this 2D colony. We already view the merged view where you can see the 2 different colors. This looks actually quite reasonable. And this will be the processing of this colony will now also be much faster than the example that we did before, because not only is it just 1 image, it's also just 1 plane, just a 2D plane. And if I look into the directory that was created, this one here, 2D colony position 1, it contains my TIFF files and it contains my metadata files. So everything has been, again, generated by BiofilmQ in the way that it is necessary to be read and to be processed. All right, so now coming to this particular image, what is it that I would want to know from this image? What is it that what kind of information can I get from there? Well, this is a prime example for how we can use the merging strategy or the merging tool of BiofilmQ to make sense of our data, to combine 2 channels and read sort of the combined information. So this is what we are going to try today. However, before we can merge channels, we first have to segment them. So we are going to do our segmentation just as we had it in the previous example of this workshop. We will not have any registration because there is no time series. We won't do any pre-processing, so I'm just going very quickly now through these steps. We will not apply a top hat filter because I discussed in the first part of the workshop, this typically doesn't make sense if I have single cell, it only makes sense if I have single cell resolution. And then in the thresholding, I can choose between different thresholding methods here because I have only one image. It actually makes sense to do a manual thresholding. And I can do this now for both of the channels. I'll start with channel 1. You can see that initially everything is considered to be part of the colony, and now I can sort of choose where I set my threshold in such a way that it makes sense. And of course, there's always a trade-off between kind of having the outline of the community, which is a bit darker, and getting rid of these dark paths in between, which are not actually part of the image. I can try to improve this by actually going back to my denoising. I can decrease the size of the average filter, and maybe it even makes sense to apply a top hat filter. However, I will still not do it in this case, or maybe we can try it with like a lower size and see what it does. And hopes that the parallel pool has to start again. Remember that from the first part of the workshop. But it should have no trouble once it has started, because again, this is a 2D image, and therefore processing happens really, really fast. All right, so now I have this image here where I have to lower the threshold quite a bit. And as I can see, actually the top hat filter really didn't help in this case, and we will therefore be removing it as originally planned. All right, so this is my image. Let's find a threshold that we can live with. Maybe something like this, 2600. And after that, we perform the segmentation. Here we have to choose a good cube size. In the previous sample data, we chose 16 pixels as the cube side length. Now we should choose the cube size according to basically the size of the smallest parts of the image that we still want to resolve. So here I would actually go lower. Maybe I would go for something like such as 8. And we can see that here, for example, of course, the cube side length will be much, much greater than a single cell, because we're not actually interested in these single cells, but in these relatively fine structures over here. Maybe we can even go lower to a cube size of 5. And the post-processing, I do want to remove a small object, which I'm going to set at 100 voxel here. And then I'm going to segment this first channel. Which seem to have worked well. And after that, we can move to the second channel and just perform the segmentation on the second channel in the same way. So yeah, let's go to the denoising part directly, to the thresholding part, and choose an appropriate threshold here as well. So here maybe we choose 2800. And then we just perform the segmentation on this channel as well. So now we have both channels segmented, and the question is, how can we put them in relation with each other? And this is where the merging comes into play. Merging to channel essentially means that the biomass of both of these channels, or of both of these segmentations, is combined, but the information which biomass pixel basically sort of belonged to which channel originally is kept in such a way that for each object or each cube that will be exist after the merge, we save the information what percentage of the cube was filled in channel 1, what percentage of the cube was filled in channel 2. And we can basically just do this right now, and then hopefully you will be able to understand a little bit more about this when we look at the result. So now we are doing this merging here, where we add the information of channel 2 to channel 1, which means that essentially, after clicking this button, these two original data files, they will be stored in a backup place, and the data file of channel 1 is going to be replaced and overwritten. What this means in terms of sort of our directory is that in this data folder here, I now have a non-merged data backup folder that contains our non-merged segmentation for both channels. And the channel 1 has been replaced, the segmentation of channel 1 has been replaced by the merged segmentation. We can look at this now in the same way that we have looked previously at segmentation results. I can load them. And now you can see, first of all, if I load them, I just look at something like volume, just to see. I can see that I have my colony, which is segmented, and this is the entire colony, because this colony either consists of pixels which are fluorescent in channel 1 or in channel 2. Technically, there could also be some of them which are present in both channels, but all of the pixels that we see in this image, they are either red or blue in the part of the colony, which is why the entire colony is sort of filled in this case. Now the two, or actually the three properties that are produced by the merged are overlap between the two channels and the relative abundances of each channel. Let's have a look at the overlap first. You can see that there's quite some overlap, and this happens in the regions in which the channels touch. If you look at this image here, we can see this is, for example, here, this region here, which is all yellow, is this region over here where we have sort of a very thin mixture of red and green. Then we have these sort of regions in here, which are in between the two channels and so on. So there's quite a bit of overlap between those two channels, and we could fix that by putting the threshold of each of the channels higher. For now, we will actually be content with that. And we will move on to look at the other properties, which are the relative abundance of channel 1 and the relative abundance of channel 2. Let's just put them next to each other so that we can see what we're looking at here. So essentially what this tells us is that everything that is yellow here means the cube is completely filled in channel 1. And then I have some, and I think this is a little bit more easy to tell in the relative abundance of channel 2 and these cubes here, and have some cubes that are partially filled in channel 2, which means that the cube is contains some pixels which are assigned to channel 2, but most of them are actually assigned to channel 1. And this is given in terms of a percentage between 0 and 100. And you can see the 0 is black. So these pixels here, there is a cube there, but it just has 0% channel 2, because all of the pixels in this cube are exclusively red and none of them are green. If I look at them next to each other, I can see that through the thresholding, I have been able to sort of separate these two different kind of parts of the colony, the red and the green one, but there is still quite a lot of overlap. So I could go back now and I could adapt the threshold and see if I can fix this. I think for now, actually, we will keep it this way because we want to look at a few other things during this workshop. But of course, you can play around with the threshold as much as you wish. Now, the second thing, of course, that we always want to do is the parameter calculation. So here the question is, what actually are the relevant parameters? And of course, this filling or the relative abundance that we just looked at is the parameter that we are interested in the most. Although we need to have this in some form of spatial context. And this is where we want to get the spatial context by looking at the distance to, for example, the biofilm center or the surface of the biofilm. You can see that from our previous analysis, there are still some parameters checked in here. So I'm just unchecking them now, just all of them to be safe. But overall, this is fine. We basically just want to have these two properties measured, the distance to the center of the biofilm, and then the distance to the surface. And the distance to the surface we also set at a lower resolution of 5, which in this case is the cube side length. All right, I can now calculate both of these properties, which should be relatively fast because I have only one image in 2D. And once they are calculated, I can have a look at them and see how they make sense. So it seems that the parameter calculation is complete. And we can now load the segmentation results again. And now I have these additional properties, distance to biofilm center and distance to surface. But I also have this distance to biofilm center at substrate, which is only relevant if I have a 3D biofilm. So if I look at the auto view of that, I can see that this is actually very nicely, like very smooth. I have nearly 0 in there, and these pixels are just black because they are very, very dark blue, but they are in fact a dark blue and not 0. And then as I go out, they become brighter and brighter. One thing I can also see is that I do not actually see, or it's actually very difficult to see the individual cubes in here. So I might actually have picked a relatively small cube size that I might want to increase in order to speed up my computation time. However, in this case, I think this will not be necessary because we have already calculated everything that we want to calculate. Here I can see that maybe for the surface, I might actually want to increase my surface resolution because I get these artifacts here, which are likely due to me having a relatively small distance to surface. And I'm just going to put 10 here and run this calculation just off the distance to surface again in order to correct for this and see if it does make any difference. Okay, so let's have a look. We should have one additional parameter here, which is the distance to surface with resolution 10. Okay, and now we actually see that it's gotten a bit worse. So I'm guessing that actually I should decrease the resolution rather than increasing it. And you know, that's also how sometimes you just have to go back and forth a bit between setting properties or setting the settings for these parameters and determine which way makes most sense. But yeah, in principle, while this is still calculating, what we have is we have these two measures, distance to biofilm surface, distance to biofilm center, that are essentially opposites of each other, as you can see. But you can also see that there is a little bit of a difference, which is caused by the specific shape of the biofilm. And that means that depending on what kind of question you want to ask, you might want to choose one or the other preferably. Okay, I think we're still calculating this property. But we could actually probably also just stick to this representation here, which is resolution 5, and live with these small artifacts at the top. Okay, but now it is actually 10. There's one last thing before we move on to the visualization part that I wanted to show you, and this is that we have something that is probably quite useful, but requires a little bit of explanation, and that is that we have this custom parameter here. So I wanted to give you an example of how we can use this custom parameter, which is essentially a combination of other parameters. And the idea that I had here is that we could look at the biofilm distance to center, but in a normalized way. Because think about if you want to compare different colonies, maybe they have different growth rates, they have grown to a different radius overall. during the same time, and you might want to just look at this in a normalized manner. So what we can do in this case is first we have to give a parameter a name. So we call this maybe distance to center underscore normalized. And then how could I normalize it? Well, I know in the center it is 0, and then it goes up to a maximum value. So I choose this property here, distance to biofilm center. And when I click add, you can see that it appears in here in these brackets. And now I can basically write any type of code that is MATLAB interpretable. So what I want to write is I want to divide this property by the maximum of this property again. So this is essentially my formula, and I can test if this formula is valid. So basically, if MATLAB would be able to read it, and it says okay here, so I can actually choose this as a parameter to calculate. And we can calculate this property right now and have a look at it to see if it is the way that we expect it to be. So in principle, it should look exactly like the distance to biofilm center, except that in this case, it should be between 0 and 1 rather than between 0 and the maximal value. So let's have a look at the distance to biofilm center as it was originally, which looks like this. And now we should have our distance to center normalized up here. And we can see that they do essentially look identical. I'm going to make it a bit larger and put it next to each other. So this is the distance to center. But if I add the heat map, or the color map in both cases, I can see that one of them goes between 0 and 1 and the other one between, well, 200 and 1400, which is a micrometer here. So now I have found a way that I can look at this in a normalized manner. And we will now move on to the visualization part where we want to visualize this data. And essentially the type of question that you would ask with this type of data is that how does the sort of the relation between the two channels or the two strains change sort of across going from inside to the outside of the colony, so basically across time. The way that we can do this is with a 1.5D histogram plot. And in this plot, we choose an x-axis, which in our case should represent the time, which means that we are going to use this distance to the biofilm center, and here we can use a normalized distance, because that makes it a bit easier to choose the range, which we are going to choose from 0 to 1. Here you can basically choose the range of your plot. And then as on the y-axis, we want to have the relative abundance of, for example, channel 1. And we do have to pick how many bins we want to have here. And I think 100 is way too, or might be too much. Let's maybe go down to 40 in this case. And we want to have the mean rather than the sum. Everything else should be okay. So let's have a look. And here I'm just removing this add plot to current figure. So yeah, this is what we get as a plot initially. Here we can see that we go all the way to 100%. This is channel 1, which would be the red channel here. You can see that red channel is quite present or is quite abundant. And interestingly, if we do channel 2 now, so going up and choosing channel 2 as my second axis, and this now I can again add to this current plot just to see how it changes. We can see that channel 2 follows a slightly different pattern. In particular, the abundance seems to drop after a certain distance. And another thing that we can see is that actually both of the abundances do not sum up to 100, but they are higher than 100%. This is because there are pixels, and we saw before that there are quite a lot of pixels that are present both in the green and in the red channel. Which means that if you would add up the percentage of the relative abundance, you go beyond 100%, but this is okay. This is not unexpected. Yeah, so this is the result that you would get from here. Of course, you can go back again and you can decide that you want to change the thresholding. You can decide that you want to change the cube size, or that you want to calculate some other properties that you want to measure over time, but this was just one example of how to handle these types of files, how to perform the merging, and then how to, in the end, also define your own custom property, which we did with this part here. So that would be the slightly shorter second part of the workshop. Overall, I hope that you enjoyed the workshop. I hope that you learned a bit. This will all be available on YouTube for a while, so we can watch it later on again. There are also a lot of other tutorials on YouTube, as I have mentioned in the introduction, and I would be happy if you checked some of them out. Also, if you reach out to us on the image.sc forum, where we are delighted to answer any questions or to discuss any questions related to BiofilmQ that you might have. But yeah, for now, I'm at the end of the workshop. Thank you for sticking with me, and hopefully see you around the I2K, or see you around at the forum or at any other platform. Bye.